All right, so we've now covered the four classical areas. I shouldn't say classical, I'm sorry, the four main areas of modern genetics, being transmission genetics, population genetics, cytogenetics, and molecular genetics. So in no particular order. If I were to ask you to put it into an order, I would ask you to go from macroscopic to microscopic. And that would be population genetics being the most macroscopic, then comes transmission genetics, then comes cytogenetics, and then finally we get to the molecular level, molecular genetics. Now going back to this, so transmission genetics, let's talk a little bit more now that we've seen what to expect. Let's talk about how this works. Right. So you've seen the slide before, right? Transmission genetics. Geneticists use experimental organisms to study how traits are passed from parents to offspring. We talked about fruit flies and E. coli and yeast, etc. And so now let's talk about what is a good organism to use. These are the 6 examples of different organisms, very different organisms, right? You get from a plant to a soil worm to bacteria to a mammal. And so essentially you have very different organisms here, but they do have certain things in common that make them ideal for being used in transmission genetic studies. So what they're called sometimes is a model genetic organism, but a model organism, you have to be very careful when you use that. So a safer way of calling this would be an experimental organism. When you say experimental, you just mean that you can use it for something. Model means that it has more of a connotation to it. Now these are examples of model organisms for certain things, but let's just talk about what is a model genetic organism. What's a good organism to use in transmission genetic studies? And so organisms with traits that make them particularly useful for genetic analysis, I should say, those would be model genetic organisms. And so number one, in today's day and age, we want to make sure that we've had the genome sequenced. So all of these organisms have had their genome sequenced for quite a while now. I think I may have mentioned that C. elegans was the first animal to actually have its its genome sequenced. But it's not just that they have to have their genome sequenced. There's more to it than that. So what you're looking for is a short generation time. So obviously bacteria actually gets so short in generation time. E. coli can double in logarithmic phase every 20 minutes. So it's a very short generation time. So that actually might be on too short. But mice are manageable. All the organisms I showed you on the previous slide are manageable. Human beings, on the other hand, not. Because obviously we've already talked about how we're not going to do any controlled matings or anything like that. But even in the situation where that might occur, it's not feasible. It's not realistic because the generation time is, again, not just 9 months of gestation, but we're talking about years and years and years before. So think of it this way: before you get to the next generation, right? When a baby is born, there are many years before that person is going to reach sexual maturity and then actually reproduce. So you have to add that time in as well. So that is definitely not a short generation time. We could be talking on the order of 20 years. Obviously, it could be a little bit less than that, but then again, it could also be quite a bit longer than that. So what we're looking for is a short generation time with these organisms. We want manageable numbers of progeny. And you might be thinking that, like, wait a minute, E. coli is not going to give us manageable numbers of progeny because bacteria, there's going to be just countless bacterial organisms there, cells. But in fact, actually, we could do certain things to basically select for the cells that we want in our study, right? The cells expressing a certain gene, we can couple that to, say, for example, antibiotic resistance, and then the antibiotic placed into the medium will kill off all the other cells that we don't care about, and then we just keep what we care about. And so that's actually a manageable number of progeny, or mice for that matter. They don't just have like one, mouse pup per litter, right? There's multiple mice per litter, and so you have a manageable number of progeny. Okay, in addition to manageable numbers of progeny, we are going to look for adaptability to the laboratory environment. So all those organisms I just told you about on the previous slide. So we saw mice, we saw E. coli, we saw yeast. Yeast are very manageable in the laboratory, very adaptable to the laboratory environment, very easy to work with in the lab. And then of course, and I'm biased, right? Obviously, I did my graduate work in a yeast geneticist's lab. But while I was in graduate school, I also worked with C. elegans, and I also worked with MDCK cells, so Main Darby dog kidney cells. I think I almost said doggy, but they're dog kidney cells. That was cell culture. But all of those are adaptable to the laboratory environment. I'll give you an example of something that's not, unfortunately, not adaptable to the laboratory environment. So there's big money in what's called farming with a pH, right? So you may have heard of biopharmaceuticals or the pharmaceutical industry, but farming meant to sound like farming, like, out in the field, tractor and all that sort of thing. But with a pH is really looking for certain compounds in nature that are of therapeutic value that would be wonderful if we could reproduce them in the laboratory if we had an organism that made them, that had a short generation time, manageable number of progeny, and adaptability to the laboratory environment. But there are some organisms that are very valuable because of what they produce, and yet they are not adaptable to the laboratory environment. The perfect example would be a sea cucumber. If you've ever heard about or are familiar with a sea cucumber really is if you don't know how they survive, then it is an absolute enigma on how on earth this thing has not gone extinct. So sea cucumbers are, literally they're named after food. They're just sitting there at the bottom of the ocean. It's a cucumber at the bottom of the ocean. You would think they would be eaten and they would have gone extinct long ago because they're basically a buffet, but they're not. So the thing about them that you should know is that they do not have they don't have teeth. They can't swim away. There is no fight or flight or anything like that. They're just there. They're just like basically kind of blob like little structures on the bottom of the ocean or in the ocean floor. But the one thing they've done is they have adapted and they have basically made for extreme survivalists. What they do is as they are consuming the different organisms and microorganisms and the chemical compounds in the organisms that they consume, they actually use those. And so they've resorted to defending themselves through chemical warfare. And so they make some of the most complex chemical molecules humankind has ever seen. And they are far more complicated than we can easily synthesize in a laboratory, and they have tremendous therapeutic value when used in moderation, because they've been evolving for millions of years, and they've been there. They are masters of chemical warfare. Let me just put it that way. But they are not at all adaptable to the laboratory environment because they're down there at the bottom of the ocean. Try and simulate the conditions at depth, right? At deep ocean depths. So we're going to have tremendously cold environment. We're going to have a very extremely high pressure environment. As soon as you harvest one of these sea cucumbers and then bringing it up to the surface to try and grow it in a laboratory or anything, just removing it from the ocean floor, by the time you get to the surface to put it into a high pressure environment, you've already decreased the pressure and the thing is basically just, expanded, exploded, and died. But they're very interesting. We get all sorts of these compounds from ocean creatures because they've been around for a lot longer. One that's actually on the market now is called Prialt. So Prialt is used as a pain medication for severe pain. We're talking burn patients in the hospital, and the compound was isolated from the Australian cone snail. And so my younger sister travels the world, and basically she doesn't really have a job. She just travels the world. And so I'm sure that's not how she would describe herself, but she's my younger sister, so obviously I can't say anything good. But basically, so she is, there was a while ago where she was going to go scuba diving off the Australian Great Barrier Reef, and I told her, hey, oh, that's great, just don't touch anything. And she thought I was kidding. I'm like, no, I'm serious, don't touch anything, because the Australian cone snail, for example, if you were to touch it, you wouldn't even know before, you wouldn't even have the sensation of touching it, because the Prialt, Prialt is the trade name for the drug that's now used, but basically the compound from which Prialt is made will just absolutely freeze your your central nervous system, right? It's a neurotoxin. And so you won't even know what happened. It's over, right? You are basically frozen, you're dead. And I mean, we're talking on the order of milliseconds. And so I believe Prialt has the potency. So it's administered to burn patients in, like heavily diluted, 1 part per billion or whatever, of the original amount that the cone snail would actually inject into you if you touched it. And it has like, I think 100,000 times the potency of morphine. So it has to be administered very carefully so that, you don't just kill the person with, an overdose, but it's a very effective analgesic or pain reliever. So what we're looking for in these model genetic organisms is a short generation time, manageable progeny, and manageable number of progeny, adaptability to the laboratory environment. And so then when you say adaptability to the laboratory environment, you also want to be able to house them and propagate them inexpensively. Because let's face it, I mean, it's all going to come down to whether you can afford to keep these animals or organisms, for that matter, alive, right? So, like in research facilities, usually mice are very commonly used in biomedical research, and so most institutions have a mouse house, right? And that's not just me trying to be cute. That's actually what we call it. So everywhere I've worked before. I worked at Lawrence Livermore National Laboratory. I've been at a number of different institutions, UC Berkeley, et cetera. We just call it the mouse house. Everybody calls it the mouse house. And so that's where, you have some of the animals in your laboratory, but then generally they're housed in the animal care facility. And so they have to be housed humanely, for obvious reasons. They have to be housed humanely. Experiments that are going to be done on them have to minimize any sort of pain, minimize, any sort of suffering. If there is any sort of pain or suffering, then it has to be, heavily vetted whether that experiment is absolutely necessary. And so all of this takes money, right? And so if you're dealing with an organism, say, for example, like trying to bring a sea cucumber up from the bottom of the ocean, you have to have it in a high pressure environment, very cold environment. And then on top of that, like it can't be, I'm sure it's light sensitive. And then on top of that, how many are you going to actually be able to have? It's not realistic to try and consider that as a model genetic organism. That is, and unfortunately for us, not a model genetic organism. However, zebrafish are a great example of all of these. All right, so basically the value of a model organism is illustrated by the identification of genes that ended up affecting skin pigmentation in humans using zebrafish, right? Regular old zebrafish. You can get zebrafish at pretty much any pet store, and they are very common, and they are also very good to use in a laboratory setting. So here's the thing about them. So they have a short generation time, and they have, manageable number of progeny, and they are extremely adaptable to the laboratory environment. You would think you're actually being inhumane if you walked into a zebrafish laboratory, because they like to be in very high density. So you look into this fish tank, and it's just a writhing mess of zebrafish. You can't see through the tank or anything like that. You normally like to, remain in very large schools. And so when you keep them in a tank, you want to have a high density of organisms in the same tank, so that reduces their stress. But the thing about it is that I'll show you an image here in just a second, but think of them as like a dark gray color, right? And then for some reason, like a charcoal almost color. And then for some reason, in any zebrafish tank with that many fish in it, there's always these, like a few of these albino variants, right? So they're not totally albino, but they're what are called golden mutants, because they have sort of a pinkish goldish, I don't know. They're certainly not dark gray. I'll show you an image, but the image I'm about to show you, the reason why I'm saying it before I show you the image is because the image is taken under a tremendous amount of light, and so they don't really look like this. So let me show you what I'm talking about. Okay, so what we have is a normal zebrafish. I said to, imagine it as like a dark gray charcoal color. You could certainly see that in the lines, but then you're like, wait a minute, this looks like yellow and black, right? But again, remember, this is a fish that has been taken, an image that has been taken of it under a light microscope, where there's a tremendous amount of light that is, just shooting through it. So generally speaking, they are going to be sort of a dark gray silvery color. And then this one, you can see, kind of has like almost like I don't know. It's called the golden mutant, but it's almost like a peach sort of tone to it. You can see it better in this image. Like, that's kind of what it looks like. And it really stands out in a tank full of basically what appear to be, under normal light conditions, silver and black fish. And so what I was saying a moment ago is that we can use many different organisms such as zebrafish to learn about other organisms. Most importantly, what we care about the most would be to learn about human beings. So zebrafish provide a great example of this. So we learned a lot about skin pigmentation from zebrafish. Here's what happened. So let me tell you the story of zebrafish. So we've got basically here is zebrafish, right? And here's the golden mutant. And finally somebody was like, why is there always a golden mutant in my tank? And they wanted to know why that was happening, how that occurred. And so they started looking very closely scientifically at the golden mutant. What they found very quickly was that these are what are called melanosomes. And you can see here's a little arrow pointing to a melanosome in a normal zebrafish. Here's an arrow pointing to those little tiny dots there are also melanosomes in the golden mutant. As you can see, these melanosomes, so there's your scale, right, your scale bar. So as you can see, these melanosomes are quite a bit smaller in the golden mutant than they are in, and they do have some that are close to normal size as well, but they have quite a few that are teeny tiny. So they're quite a bit smaller. Generally speaking, the melanosomes are generally quite a bit smaller than the normal zebrafish. And so they thought, oh, okay, well, maybe it has something to do with the melanosomes. They weren't really, specifically looking for skin pigmentation in humans at this point. They were just trying to determine why the golden mutant was your almost, you're far less pigmented. Let me go with that. You're far less pigmented variant of the normal zebrafish. And so what they found through some studies is that the golden mutation was recessive. So recessive means that you have to have two copies if you're diploid. You can't have a copy of anything that's dominant. Dominant would mask something that is deemed recessive. We'll get to that a little later on when we start talking about classical genetics and Mendelian dominance. But for right now, let's just say it's a recessive golden mutant. So the recessive golden mutant is lighter. It has fewer, smaller, and less dense pigment-containing melanosomes. Those are those little dots I was talking about. And so they thought, okay, why? Why does it have fewer and less dense melanosomes? And so what they did was they did a little biochemistry, and they found they were able to clone the golden mutant gene. And from some biochemical analysis of that gene, they characterized the gene, and they found that and I'm glossing over a lot of this because this is just a quick example. We'll go into much more detailed examples later on in the course. But researchers found the cloned golden mutant gene and then found that it was involved in calcium uptake in melanosomes. So that calcium uptake is absolutely necessary for the melanosomes to function properly, to grow and get larger and be able to make the pigmentation that you see in normal zebrafish. All right, so now they've cloned this gene that is responsible for the lack of pigmentation in the golden mutant of zebrafish. Zebrafish are vertebrates. They are phylum Chordata just like we are. And so, therefore, you could just, well, now you could say, oh, well, let's just check, since we have the human genome, let's just take this gene sequence, and then let's just do a blast, or basically a basic logarithmic algorithm search. And we could just essentially take the sequence and blast it against the human genome, see if anything matches. Because remember that these genes are, at least, very often they are thousands and thousands and thousands of bases long. And so that's 1,000, let's just call it like, I don't know, 400 kilobases, right? So 400,000 bases. And that's, it could be larger, it could be smaller, it could be much bigger, it could be much smaller. But basically, if it was 400,000 bases, remember that's a unique sequence. It's 400,000 bases in a particular order of A, C, G, A, A, T, H, C, C, A, G, G, A, T, H, C, something like that, right? And so the likelihood of there being something similar, a 400,000 nucleotide sequence that was similar by random chance, is basically, that's not going to happen, right? You're basically going to find the human variant, the human analog or homolog to the zebrafish gene, if you get any sort of results back. And guess what? They found something. So what they did was then they found a human homolog, right? So this is just the designation given to potential genes when the human genome was sequenced. So SLC24A5, right? Nothing special, no name yet. And so then they found that gene, and then that's about that's a lot of work, and so maybe they published that, and then other scientists picked that up and they went to see what the correlation was, etc. And so later it was determined So later it was determined that if you examine human populations, so this was all, we did some transmission genetics, we did some molecular genetics to figure out calcium uptake and the function of this particular gene product. We did a little bit of, in silico work where we like basically searched the gene against the human genome. Then they examined human populations, so you had to draw from population data, population genetics data. And what they found was that SLC24A5 appears to be responsible for, and look at this number, 24 to 38%. So that's greater than a quarter of the reason for the differences in pigmentation between people of African and people of European descent. Because obviously we generally have a very different, skin tone between people of African descent, people of European descent. But upwards of even a third, right? So like a quarter to a third or more of the difference is responsible, or this gene is responsible for that. So we learned from zebrafish. So somebody looking at a fish tank being like, why is that one kind of pink, right? Going from that to actually figuring out how humans are pigmented, their skin pigmentation, how that occurs. Or at least gives us insight. I mean, it doesn't tell us the whole story, but it certainly is a large chunk of the story. So this is an example of how other organisms, these model genetic organisms, can be used to help us understand, other organisms' genetics, even human genetics. So now let's go back to the idea that, and I've said all of this aloud, but as I said, I foreshadow, right? So here are some bullet points talking about how genetics is not new, right? And so remember originally my question was, is genetics a new field of study? And the answer is yes and no, right? So yes, if you're talking about molecular genetics, no, if you just say genetics, all of it, right? So genetics and even biotechnology. So don't think biotechnology is just all that molecular stuff of cloning and subcloning and stuff that we do nowadays. Biotechnology, the example I gave you earlier of the farmer that chose to grow that corn that turned out to be drought tolerant last season, this season is going to grow basically just that type of corn. That's an example of biotechnology. That's a human being utilizing life for some sort of purpose, right? So that would be the whole biotechnology. So we're producing food, but we are selecting what traits we're looking for. That's a rudimentary form of biotechnology. So genetics and biotechnology is, about, well, we could say 10,000 years old. Might be older, but 10,000 years old seems like a reasonable number, because that's about the time where we could say where we first arose, genetics first arose when ancient hunter-gatherer people that were just scrounging for what they could find in the environment switched to agrarian societies. As soon as we started domesticating livestock and crops and things like that, we started breeding, we started choosing, what livestock, what crops we wanted to grow, and making crosses, and even cloning plants back then, because it's so simple and can be done, that even Aristotle wrote about the cloning of plants, because it's been done forever. I think I may have said this earlier. Plants don't scream, plants don't run away, so nobody's really had a problem with us messing with plants for a very long time. So when people settled, they domesticated plants and animals, as I said. And so you can see here's cuneiform describing, actually depicting, basically biotechnological techniques in farming. And then we have a number of different grains and wheat where these are heavily, even back then, these are heavily modified genetic stocks. Let's take the ancient Egyptians, for example. The ancient Egyptians are absolutely amazing in many ways. Their technology was so far ahead of any other civilization at the time. And one of the things they had, and this is absolutely true, is, and we have all sorts of facts to back this up. I can tell you what those are in just a moment. But one of the things they had was a genetically pure strain of yeast that they used. So I believe it is Schizosaccharomyces pombe, I believe. And that genetically oh, no, I'm sorry. Saccharomyces winlocki. That's what it was. So Saccharomyces winlocki. And so that strain of yeast is a genetically pure strain that the ancient Egyptians cultivated and used to make beer and bread. And the reason why we know this is absolutely true is because, yes, you can find some hieroglyphics depicting, what they were doing. But more importantly, spores, yeast spores, fungal spores can last an extremely long time. They are extremely hardy. They can survive in the vacuum of space. They can survive re-entry. They can survive. And so when you find fungal spores in crypts and tombs and pottery that has been sealed up, hermetically sealed in pyramids for a very, very long time, those are viable spores that actually were recultivated. And so nowadays you can buy like beer made from Saccharomyces winlocki, because and it's marketed as like ancient Egyptian beer, because those spores will survive that long. So we know that they not only wrote about it, they depicted in their hieroglyphics that they had a genetically pure strain of yeast that they used for baking and brewing. But we also have the strain. The strain survived all those years, sealed up. So the farmer that selects a particular strain, or the ancient Egyptians that chose to use Saccharomyces winlocki, they all are going to exhibit examples of I believe that's kind of redundant, but they are basically giving us examples of artificial selection. And so artificial selection is where you collected seeds or tubers or seeds from plants with the most desirable traits, or you breed only prized animals. That is artificial selection. That is a form of biotechnology. So we've been doing that for a very long time. So this goes full circle back to, you're not going to find any sort of food that is not genetically modified, because if it's been cultivated, then like to find like an ancient grain or something that you happen to like go deep into like the Amazon rainforest, and you find something that no human being has seen forever, right? Like has ever seen, then okay, that would be a food. If it's a food source, that would be a source of food that has not been genetically modified. But pretty much everything in our food stream has been genetically modified, because obviously we've been cultivating our food for quite a while. So the material that you keep from these things, like the seeds and whatnot, that's called germplasm. And so germplasm is something that has been traded forever. Ancient farmers preserved seeds, cuttings, whatever it may be, and they even traded these genetic stocks. And germplasm is actually in the news again. You can look on our PolyLearn site for this section, right, chapter 1, and you'll see the Svedberg seed vault, right? So we have I don't know. How about this? Let me say this. If you haven't seen the movie Interstellar, then do a quick little like read a quick synopsis on it, and then this will make a lot more sense. So in that movie, I enjoyed the movie, but in that movie there is a premise where humankind needs to leave Earth, because there's a particular, I think they call it blight or something. There's a particular like fungus or disease that is consuming everything, and so crops are dying off. And this particular organism feeds off of nitrogen, and that our atmosphere is mostly nitrogen, and so it is, it's spreading rapidly, and so it's basically killing off different types of crops, and so people are going to have a hard time cultivating food on Earth anymore, and so they need to leave, right? That's part of the premise of the movie. And so that's not realistic, number one, because it's so hard for organisms to fix nitrogen from the atmosphere. Like the nitrogen fixation is an entire section of biology, biochemistry, whatever. And so, but it's also, the other thing is that there's no mention of all of the germplasm that humankind has stockpiled in seed vaults around the world. So we are in a doomsday scenario preparation all the time. We are, those things that go extinct out in the wild, we still have cuttings and seeds and germplasm of those organisms. And so we preserve germplasm today as insurance in doomsday seed vaults. But climate change is threatening it. So if this was a normal lecture in class, I would ask a bunch of questions of my students, of you all, and see how many people have heard of these. And it would be really wonderful if I could actually show you this, but you could look it up. Look up the, you'll see it in PolyLearn. Look up the Svedberg seed vault. And what's really dramatic is that this one, I can't remember. I believe it was in Was it in Greenland? I think it's in Greenland. But it basically there was a seed vault that was built into the side of a mountain underneath a glacier, because that would help with refrigeration, because it would naturally be kept cold, and wouldn't need to be heavily reliant on electricity to refrigerate all that germplasm. And we're talking, when I say refrigerate and freezing, we're talking deep freeze. We're talking very, very cold temperatures, right? So a refrigerator is usually at around 4 degrees Celsius. Your regular freezer at home is at negative 20 degrees Celsius, and biological tissue oftentimes is stored either in liquid nitrogen or at a negative 80, a negative 80 degree blast freezer. So what's really sad, and what this link kind of takes you to, if you were to click it, so Norway, sorry, it was in Norway, but basically it takes you to, a story about how they had to retrofit it with all sorts of refrigeration and backup systems, because the seed vault, that you're not allowed to know where it is. You're not supposed to know where it is. You can't go take a tour or anything like that. These are under lock and key. These are high security facilities. But this one's just out in the open now on the side of this beautiful hillside that's like sitting in a lovely meadow, because there's green grass going everywhere, because the glacier that was, over it has receded so far back because of climate change that it is no longer in a deep freeze, right? And so now it needs to be artificially kept cold, right? Which is just another reason why climate change is not good. But that's besides the point. The point is, germplasm is the material, the genetic stocks that we keep. And humankind, at least, one good thing to remember is that all over the world, there are caches of these just tremendous amounts of germplasm for different crops. So we could start over. So honestly, there are many scenarios in which these seed vaults were made, but some were, as a result of the Cold War and the idea of like a nuclear winter that would obliterate, like crop plants. How would you start over once, the situation, the conditions became viable again to grow plants and farm? And so you had to have germplasm stored for that type of scenario. And just to really drive home the point, here's an image of the Svalbard seed vault. Forgive my pronunciation. I know it's spelled Svalbard, so Svalbard, I would think. I may have said Svedberg in the past. That's because that's a unit of, what is that, sedimentation? But the Svalbard seed vault, let me try that again. The Svalbard seed vault. So here's what it looked like back when things were going really well. And then, humankind happened. And so here's what it looks like now. So you can see it's not so much in the snow anymore. And so if you need yet another reason to be worried about climate change, well, let me just add that to the list. So I said that genetics is not new. Let me give you some examples, right? So the ancient Greeks, like Alcmaeon, so this is circa 520 BCE, right? Believed in the concept of pangenesis. So let me back up a second. Remember how I said that genetics is not new? The idea, if you define genetics as the study of heredity, well, then that goes back as far as humankind goes back, because like I said, the very first humans, no matter, even before we had written history or anything like that, they knew that if you had a child with somebody, that child was a little bit like you, a little bit like the other person, families, right? And so then people came up with explanations on how we pass on our traits. And so some were based off of, religious beliefs. Some were based off of logic, right? So the ancient Greeks were very big on logic. And so they didn't, scientifically determine this. But let's talk about some of these older concepts, and then we'll bring it full circle to what is actually occurring now. What we know is actually how we pass on our traits. So Alcmaeon put forth the idea of, or believed in the concept of pangenesis. Pangenesis would be absolutely awesome if it was true. But the fact that you are in school is proof that pangenesis is not how we pass on our traits. So in pangenesis, the idea is hereditary information travels to reproductive organs before being passed onto the embryo. So could you imagine if we had all the knowledge, all of the learning that has taken place in not just our parents, but our grandparents and every generation before us? Could you imagine how advanced our civilization would be? Everybody would start with basically all of the knowledge of humankind at birth, right? You wouldn't have to learn anything. And so obviously that's not how this works. But back then they thought that that pangenesis was how this worked. So information that's learned would travel to your reproductive organs and then be passed on to your organisms. So you can see that according to pangenesis concept, genetic information from different parts of the body. Genetic information being like hereditary information, like what you've learned, your memories, all that sort of stuff, passed to the reproductive organs and then transferred into gametes. The gametes, eggs and sperm, are going to end up in the offspring. And what's silly and what's unfortunate is that there are still many people today that think this is how this works. In fact, you can find almost any like genetic engineering plot in a movie, and there will be all sorts of craziness. Like I am just thoroughly disappointed in Hollywood because, they consult with individuals that actually know the science, and then they're just like, yeah, but it makes a better story to do it this way. So there's you've seen this before, like how something that's cloned has the same memories as some other organism or whatever it was cloned from. Like if you clone a person in a movie, then they can remember things, and they're like, oh, I'm getting a flashback of this, and that's just pure. It's erroneous. Is a nice way of saying it. Oh, I don't want to start this slide over and re-record. So, at least I caught myself there. Anyway, so hereditary information travels to reproductive organisms before it's passed on to the embryo. That would be pangenesis. And so ancient Greeks believed in the inheritance of acquired characteristics. Pangenesis is the inheritance of acquired characteristics. Traits learned during one's lifetime are passed on to one's offspring. And again, this idea from 520 BC has been propagated even until now, even today. I mentioned the eugenics movement, and I talked about a boat builder, right? The boat building gene. And so even today, I told you about the gentleman that I had the I won't say pleasure of listening to for quite a while about telling me how everything's genetic. People still today believe that you pass on things you learn. And in fact, that's not the case. So it's incorrect. But like I said, the same ideas have persisted through the 20th century. And, now we're in the 21st century. And in fact, people still believe that, you pass on things you've learned.